In this video, we're going to be discussing the tissues associated with lab exercise 3. This week in lab, you'll be looking at tissues associated with both epithelial and connective tissue classes. This is the first of 2 videos you should watch prior to lab. In this video, we'll only cover epithelial tissue. The second video will cover connective tissue. Epithelial tissue is all avascular, meaning it doesn't have blood vessels, and epithelial tissues have 2 major types of arrangements. Sheets and membranes, which means this kind of tissue is important for covering surfaces of the body and organs, lining the insides of bodies, organs, and it helps with protection, secretion, excretion, absorption, filtration, and sensation. The other arrangement is going to see these epithelial cells grouped into cords, follicles, and tubules, and these are mostly associated with secretion or excretion of wastes. When you begin to name epithelial tissue, you're going to do so using 2 different types of criteria. The first is going to be its structural classification of simple or stratified. This deals with how many layers of cells make up the tissue. Simple epithelia is a single layer of epithelial cells. In these, you'll see that they're all in contact with an acellular and cellular component of a basement membrane. If it's referred to as stratified epithelium, then it's going to be composed of 2 or more layers, and only the bottom layer, what we also call the most basal layer, makes contact with the basement membrane. You'll also refer to the names of epithelial tissues based on the shapes of cells that are present. Squamous cells are flat, cuboidal cells are square or cube-like, and columnar cells are taller than they are wide, like columns. In the first example that we look at, we see simple squamous epithelium. Please note that with each of these images, you'll see the total magnification at which the image was generated. So this was 400 times magnification, so this is zoomed pretty closely in. Now, most epithelial cells, when in the body, are very tightly packed together with no space between them. This particular slide is made by scraping the inside of a cheek in your mouth and then spreading the cells on the surface. So the cell junctions have actually been separated from one another. But this image is still useful because it shows you the basic anatomy of a cell. So here you could see the cell membrane or the plasma membrane going all the way around the outside of the cell, a dark nucleus on the inside, and then all of the cytoplasm that runs through the inside of the cell. This usually picks up a purple or a pink stain that's relatively light, and the nucleus stands out because it usually picks up a darker stain. The nucleus in these cells looks very nice and round, but it could be oblong or oval, depending on what kind of tissue you look at. When you look at a second slide for simple squamous epithelium, you're going to be looking within the kidney. And when you focus primarily on the outside of the kidney, you're going to see structures referred to as glomeruli. Now, these glomeruli are balls of capillaries. And if you're reading your description in the lab manual, you'll find that simple squamous epithelium helps with diffusion across capillary walls. And so all of these are individual nuclei of single, simple squamous epithelial cells. Now, because they're all grouped together here, it's a little bit difficult to see the idea of one of these cells being very flat. And so if you focus here on the outer rim of the capsule, right there, you can actually see a single layer of flat, very skinny, simple squamous epithelial cells, and they're demarcated by their dark nuclei. Right? And so you're seeing very thin, single layer of cells that form the wall of this capsule. And so both right here around the wall and then here in the middle are examples of simple squamous epithelium, a single layer of epithelial cells adhered to a basement membrane. You get a preview of what we'll look at next by looking right beside this at part of a kidney tubule, and this is showing you what we call simple cuboidal epithelium. So we'll look at the next slide, and you'll of course see this description in your lab manual to actually talk about simple cuboidal. Now, because it's simple, you're still looking at a single layer of cells, and you're going to be looking at the cells which form tubules right here. So you'll see a number of tubules at 100x magnification, and just so you know, all these tubules are separated by connective tissue. And so to see this more clearly, you're going to zoom in to a single tubule. So forming this tubule, you see first off that these cells are no longer flat and skinny. Instead, they're now cube-like. So that's the hallmark of cuboidal tissue. Now, it is still a single layer of cuboidal cells that make the wall of the tubule, And you'll notice, of course, characteristic of epithelial tissue, there are no spaces between the neighboring cuboidal cells. This is going to prevent leaking of the contents of the tubule past the cells. As with any hollow tube, you're going to see the hollow space here is the lumen. Since this would be a kidney tubule, you'd see the early stages of urine passing through this tubule. I have gone ahead here in the slides and shown you the marking of your cell or plasma membrane, along with labeling the nucleus. That way you can identify individual cells. So that's simple cuboidal epithelium. As you move into simple columnar epithelium, you're still going to be looking at single layers of cells, hence simple, but now the cell shape begins to change. To give you perspective, the slide you're studying is a cross-section of the small intestine. And the small intestine's wall on the inside is going to be textured by giving off a series of finger-like projections towards the inside of this hollow organ. These are referred to as villi. The epithelium we want to see is going to line these finger-like villi, and so you'll find the villi and then zoom into those. So here, as we take a closer look at a single villus, you can see the dark nuclei of this tissue. Right? So first off, this shows you that the nuclei are now oblong or oval. This is different from the very rounded nuclei that you saw with simple cuboidal epithelium. The other thing you might notice here is they tend to line up in a straight line. Well, since these cells are uninucleate, which means they all have a single nucleus per one cell, you can assume that this, following my pointer, is the border or the cell membrane of a single columnar epithelial cell with its nucleus inside. So that logic then helps you to understand that you have multiple cells sitting side by side with no spaces between, and with their nuclei all being at the same level, it helps you to see that this is a single layer of these columnar cells. The cells run along this direction, and when they reach the end of the villus, they'll curve back around and run back in this direction towards the outside of the organ. Now, as with all epithelium, these epithelial cells will be bound to a basement membrane with connective tissue separating those regions. And so the connective tissue here is referred to as lamina propria, one's own layer. As you look more closely here, you'll see that epithelial cells are not the only cells present. Periodically, you'll find goblet cells, and these are shaped like wine glasses or goblets with what looks like the wide rim of the glass here, facing the more apical part of the cell. And the stem of the glass would reach down in this direction, a skinnier part of the cell, to anchor there at that basement membrane. Goblet cells are responsible for mucus production here within this tissue type, and they'll lay that mucus on the surface of these cells. So of course, as you begin to study the cells, you'll see the terms apical and basal here on this slide. The apical surface of epithelial cells is what makes contact with the lumen or the space here in the organ, and the basal surface is what's anchored to the connective tissue and the basement membrane. As you continue on, you see the next type of epithelium, and this is going to be referred to as pseudostratified ciliated columnar epithelium. Now, the slide you're going to use to see this is titled the trachea or the trachea and the esophagus. So look within your slide boxes to see the correct slide. If you're viewing the trachea and the esophagus slide, you're going to have both organs on one. Be sure that you're focusing on the trachea to see the epithelial tissue that we're studying at this time. A cross-section of the trachea is going to yield more than one type of tissue. The epithelium you're studying runs right along here, and it's going to be the tissue that faces the hollow lumen. If you move superficially from that epithelial layer right there, you're going to also pass through a layer of connective tissue, also known as the lamina propria. Below that, you're going to see a layer known as the submucosa, which contains glands. Then that's going to be followed by a layer of hyaline cartilage connective tissue, which you'll study more of in the second video, followed by an outermost layer of adipose tissue or fat tissue. Again, a type of connective tissue you'll cover later. You want to make sure that you zoom in on the epithelium to study its characteristics. So we go from 40x magnification here to 100x. Here you get a better idea of what that pseudostratified ciliated columnar epithelium would look like and how it's anchored to connective tissue. Once again, true to all the epithelial tissues we're studying. We're going to zoom in one more time to get a better look at this epithelial tissue. So this is still referred to as columnar in shape because if you begin at the basement membrane, what you would see, this is not as clear as what we saw in simple columnar, but what you would see is a cell that is tall, right, and narrow and not wide. Now, because the nuclei look like they're in different heights, it does appear that this is not a simple or a single layer of cells. It is, however, just one single layer of cells. Some of the cells are taller than the others, so their nuclei sit up a little bit higher. Because it gives you this falsely stratified appearance, it's referred to as pseudo or falsely stratified. So it's truly simple, but it looks like it's stratified tissue. Now, of course, you'll see the term ciliated in its name, and that's because on the most apical surface of these cells, you're going to see cilia, small projections on the surface of the cell that increase the motility of substances across the surface. Since this is the trachea, this is responsible for helping to move mucus and debris across the surface of the cells so that it clears out your respiratory pathways. Be sure that you can identify this tissue at all three magnifications, so study it pretty intensely in each position. We move to the last type of epithelium here, and this is going to be transitional epithelium. I apologize, it's not the last, we have more. But transitional epithelium is best viewed here at 400x magnification. You are looking at the inner wall of the bladder, and what you can see here is the apical surface of the epithelium, and this would be the lumen of the bladder. So this is where urine would be stored. And if you cross down through that basal layer here, this is where the epithelium would end and where the connective tissue lamina propria would begin. You'll notice that transitional epithelium is considered stratified, and it has cuboidal or columnar cells here at the most basal layer. However, as you look at the cells that are at the top, they look more rounded or cloud-like. This is unique for transitional epithelium. This is interesting in terms of function because as long as the bladder is empty, then these cells do look puffy and rounded. But as soon as the bladder begins to fill with urine and it begins to stretch the wall and flatten out this region, those cells have the ability to flatten out as the organ fills. And so you could think of transitional as allowing this kind of an organ to transition from filled to empty. And so again, you're looking at the urinary bladder, but this is also seen in the umbilical cord. As we continue, we look at a grouping of epithelial tissues that are referred to as stratified squamous epithelium. So let's start there. First off, it's stratified. You can see here the basalmost layer of these epithelial cells. And of course, there's many layers stacked one on top of another, ending here with the most apical surface, making contact with substances in the lumen. Now the cells at the very top are squamous, and you can tell that here by their flattened nuclei. The cells at the bottom are more cuboidal in their shape. Now you'll notice, of course, that the name here, squamous, of the tissue, is actually just representing the most apical cells. And this is not uncommon. In stratified tissues, you do typically name the tissue for the cells that are at the most apical surface. Now what makes this non-keratinized is that the cells that are here on this epithelial layer, they lack a protein called keratin, which is responsible for hardening cells. Non-keratinized stratified squamous epithelium is going to line hollow passages in the inside of the body. And you're going to see this in the esophagus. So when you look at your trachea and esophagus slide, this is a good example of where you would find that. The slide you can look at in lab is also just titled Stratified Squamous Epithelium. The idea here being that it has very thick layers, so it's still excellent in protecting against abrasion, right, or friction, like you would see in the esophagus when you swallow food. However, because it's exposed to moisture, the cells themselves don't need to be hardened with keratin. As you move to the next type of tissue, you do here see a keratinized stratified squamous epithelium. You're going to be looking at two different slides to exemplify this type, but what they have in common in being keratinized is that the cells of the epithelium are now hardened with keratin protein. This is necessary because this is going to be making up the outermost layer of your skin, known as the epidermis. So the epidermis, the outermost layer of skin, is the location of keratinized stratified squamous epithelium. The protein keratin hardens these cells to protect you against an environment that can be dry. So unlike what we saw with the non-keratinized, this is exposed to the external environments. Now, when you look at the slide of skin, again, you're going to see more than one type of tissue. As an organ, that is the hallmark. An organ is made up of more than one type of tissue. The epidermis, again, is where you'll focus for the epithelium, but you'll notice here that this layer here below it is known as the dermis. There is a papillary layer of the dermis made up of areolar connective tissue, and then below that is a reticular layer of the dermis, and this is an example of dense, irregular connective tissue. For this video, we're going to continue to focus on the epidermis. In this particular slide, you're looking at skin that's known as thin skin. So this is found in all regions of the body except for the palms of the hands and the sole of the feet. It's thin because it actually lacks one of the sublayers of the epidermis that you would see in thick skin. So here again, you're looking at stratified squamous epithelium. And so beginning here is the most basal layer of the epithelium, and you can see the cells progress upwards to the more apical surface. Now, it's important here to stop and compare this with the non-keratinized epithelium. In the non-keratinized epithelium, in those most apical layers, you could still see nice cellular shapes with very clear and dark nuclei. However, through the keratinization process, as these cells in keratinized epithelium harden, they actually die. They extract their organelles, including the nucleus, and so they're not visible here in the uppermost layers. These layers that look the most fibrous and are the most hardened are known as the stratum corneum. Below those, you're going to see a dark purple or dark pink layer known as the stratum granulosum. Below that, a multicellular layer known as the stratum spinosum. And the most basal here is known as the stratum basal. So this is going to be a single line of dark cells. Again, this is thin skin. When you look at thick skin, from 40x magnification, you see the same things. Again, you can see an epidermis, which houses our epithelium, most superficially. You see a papillary layer of the dermis and a reticular layer of the dermis below that. In this particular image, you can also see a layer of subcutaneous fat here in the hypodermis. For this slide, we're going to focus on the epithelial epidermis. Thick skin is so named because it is thicker. You find it on the palms of the hands and the soles of the feet, and it has an additional layer when compared to thin skin. You can still see the hardened layer of the stratum corneum most superficially, and just deep to that is where you see the layer known as the stratum lucidum. It's a light line that is only found in thick skin, not in thin skin. Below that, you see the stratum granulosum. Below that, you can see the stratum spinosum, once again multicellular. And then finally, the most low level here is the stratum basal, so a single layer dividing the epithelial epidermis from the papillary layer of the dermis below. Be sure that as you identify your keratinized stratified squamous epithelium, that you are able to both identify it as thick skin versus thin skin, and that you're able to identify the individual layers that make up the epidermis. In the second video, we'll describe connective tissue, and you'll see several characteristics there that make it very unique when compared to epithelium. Be sure to watch that second video for the details before you enter lab this week.